Right, we'll give you a whistle-stop tour into what you can achieve with NMR spectroscopy, and specifically in the context of the scope of the kind of work that we do in the NMR facility at LMB. So what I want to do is start from the absolute basics, assuming that you have no prior knowledge of NMR spectroscopy. If you do have some familiarity with it, bear with us. We'll skip quite quickly through the basic principles of the technique, where the signal comes from, and what properties of a molecule affect the signal, so that we can then work back from recording the NMR spectrum and infer behavior and properties of the molecules in solution. I want to start from this very basic level and then move quickly through to some of the examples that illustrate the scope of the type of stuff that we're doing. So, what is NMR all about? It really comes from the basic principles of electromagnetic induction that we're quite familiar with. This is a couple of examples of so-called homopolar motors, where we have a power source, and below it we've got some neodymium magnets, and the current that flows through that wire produces a local magnetic field. We have electromagnetic induction that induces from the movement of charge a localized magnetic field that interacts with the magnetic fields in the magnet and drives the motors. Now, this electromagnetic induction applies not only at the level of currents through wires, but also at the subatomic level. So when you have the movement of charged particles, the electrons and protons in the nucleus, you have associated with that the generation of local magnetic fields. So inside every molecule, there is a native landscape of local magnetic fields that are a characteristic property of that molecule. When we put the molecules into an extremely strong magnetic field, we're able to extract information about the magnetic landscape that will reveal what the composition of the molecule is, that is the functional group chemistry and the covalent bonding network. It also tells us the shape of the molecule, any interactions that the molecule is making with other molecules, and the dynamics, the way in which the time dependence of the three-dimensional structure. So this is the reason we have NMR, because it gives us unique insights into the behavior of molecules, and we can extend it to molecules inside whole cells and the macromolecules, proteins, nucleic acids, whatever, that we find in projects controlling biological pathways. And so we can look in a mechanistic way at how biological pathways are controlled from a mechanistic and structural perspective. NMR gives you unique insights into the behavior of molecules that you can't get from other biophysical techniques. We have a very well-equipped NMR facility. We're not based in the main LMB building. We are about 5 minutes walk away in our own purpose-built NMR spectroscopy facility. I should say that on a clinical site, it is unnecessarily alarmist to use the word nuclear in describing nuclear magnetic resonance. If you're trying to put patients inside of a machine that is a nuclear magnetic resonance spectrometer, it tends to cause unnecessary alarm. So we ditch the word nuclear and just call it magnetic resonance spectroscopy. So we're more likely to refer to it as the MRS building. If it was looking at imaging, where instead of looking at the structure of molecules and composition, we look at the physical location in space, it was referred to as magnetic resonance imaging. And the MRI scanners are dotted around the campus at various places for clinical use. In our case, we have at the moment 1, 2, 3, 4, 5, 6 NMR spectrometers on site. We also are major users, one of the largest external users of the high field magnets at the Francis Crick Institute in London, and there are various other facilities that we have access to. So all of these machines are optimally configured for looking at proteins and nucleic acids that are labeled with NMR sensitive isotopes of carbon and M15. So you have access to all of this equipment. There is an enormous amount of technical detail that is involved in running, implementing, planning NMR experiments, and in interpretation of the data. What you want as your interpretation in the end, press the wrong button there, is some kind of structural interpretation. And so we have an NMR facility equipped with people who have that specialist knowledge, facility support staff. So your points of contact is either, if you have familiarity with NMR techniques, you can just come in and run the equipment yourself. You just need to speak to us to book some machine time. Otherwise, come and talk to us and we can help you plan, implement, and interpret the data in the context of your biological project. So what is it that you can achieve with all of this equipment? Why has LMB invested so heavily? The principal use of NMR is for organic chemistry and small molecule applications, where we're looking at the chemical composition of small molecules. Every chemistry department in the world now virtually will have access to NMR spectroscopy for characterizing small organic molecules. But in addition to the composition, we also get the confirmation of the three-dimensional shape of the molecule and the formation of multi-molecular assemblies and complexation. It gives you an insight into dynamics, the internal mobility, the time dependence of the three-dimensional structure. Also, the time dependence of association and dissociation. NMR is quantitative, so we can measure exchange rates. We can tell you whereabouts in a protein the flexibility is occurring, how that flexibility changes, and where you've got multiple conformational states of proteins, we can pick out where there is exchange between the different active states. Remember that proteins aren't simple static structures. The essential part of the functional properties of proteins is their ability to switch between different conformational states. And NMR can give you a great deal of information of which of the states are currently present and where there is exchange between the multiple forms that are in there. The principal advantage that NMR has over other forms of spectroscopy is that it gives you a separate signal for every atomic nucleus in the molecule. So we have site-specific detail around the entire molecule as to what's going on. Okay? It's a key feature of the advantage of NMR over other techniques. So we're able to map binding interfaces, define the locus of changes, conformational exchange, and structure perturbation, and we can look at the effects of post-translational modifications on a site-specific basis. We can get relative conformations of material. If one of the materials that you have in there, you know accurately what that concentration is, then we can use that quantitatively to get absolute concentrations of materials in the sample. And we can use that, therefore, to extract biophysical constants, thermodynamic constants, etc. So we can look at, for example, the solution structure. We can complement brilliantly the recent advances in AlphaFold and rapid structure prediction. So NMR is extremely slow compared with other structure determination techniques. If you have a protein, you want to know the structure, always, if the structure will crystallize, your best bet is to crystallize the sample and get the structure that way. It's really quick, in the order of hours or a couple of days, maybe, in most practical cases. In NMR, this information might take months to generate a full formal three-dimensional structure. If the protein won't crystallize, this might be months that you're prepared to invest. But in most cases, if you can get it by crystallography, you opt for crystallography first. The other alternative we have at the moment that's just come in the last couple of years is AlphaFold, where you use AlphaFold to rapidly generate some candidate structures, and then you use NMR to validate and sort of sift through any alternatives. And if there are multiple conformational states, then you can use NMR data to bias the structure towards a particular conformational state. We can look at the structure of residual structure in disordered segments that is out of the scope of both cryo-EM and crystallography. We look at interactions between proteins. We can define what their binding interfaces are. We can define which are the proteins that are actually directly interacting with each other and which are indirectly interacting via mediator molecules. We can get high-resolution images of models of dot structures. We can look at this site-specific effect of NMR to get detail around the entirety of the molecule on a site-specific, difficult to steer, but with site-specific detail, we can tell what's going on during folding processes, for example, or where we've got parts of the molecule that are selectively unfolding as a result of post-translational modification or whatever. We can get ideas on diffusion rates and hydrogen exchange rates. NMR is quantitative. We can get thermodynamic constants, rate constants, equilibrium constants. And what else? NMR has become one of the principal tools in drug discovery at the early stage of drug discovery for screening libraries of molecules with potential drug-like properties and screening for those molecules that bind to specific sites on the protein surface. And all of the major pharmaceutical companies have had an interest in using NMR for the early stages of drug development. We shouldn't get too fixated on the macromolecule side. When we're doing biomolecular NMR, we can also look at substrate signals from the substrate itself. We can look at substrate turnover and maybe a better handle on getting insights into a system. This is one of the systems that I've looked at a lot with Marianne Bientz's group in the last few years. It gives a good example of what we can do with NMR. This whole complex is far too large to address by NMR. The complexity of it for an NMR spectrometer is outside of our range. But we are able to get structural information and very good quality data on individual domains. So we can look at binary pairwise interactions of these components. So not looking at the whole complex by itself, but we build up, deconstruct it, if you like, and build up a complete picture of which are the components of this complex, this signalosome complex, that interact directly with each other and which are indirectly involved within the same complex. So it gives us the direct binding interaction, it gives us the contact surfaces, it tells us whether the complexes are stable or transient, and we should always include the idea that we have this integrated structural biology effort. So we are complementary to the other information that you can get from cryo-EM and crystallography. So what are the problems? So where do we start to run into particular difficulties with NMR? The first of them is the idea of symmetry. We can't resolve planes of symmetry. We are looking for molecules that are in a unique molecular environment. Many of the signaling complexes that we see for the Wnt signaling in enhanceosome, for example, have domain swap dimers that have a plane of symmetry in there. It's extremely difficult for NMR to pull to pieces complexes that have symmetrical elements in there. However, the symmetry may be an interesting and vital constraint for elucidating what your model is. So, for example, in the elucidation of the structure of C60 Buckminster fullerene, one of the constraints, the key constraints, was that the NMR spectrum, the carbon NMR spectrum of these Buckminster fullerene balls, was that all of the carbons are in an absolutely symmetric environment. So you get one single carbon signal for Buckminster fullerene, and you have to then produce a ball in which all 60 of those carbons are in an identical environment. So the symmetry component was an essential part of working out what the structure of it was. Heterogeneity is exceedingly difficult for NMR. If you've got heterogeneous samples, NMR sees everything that is in the solution, and we're really looking for very pure individual components if we can get them. And to get high resolution data, we need pure material that is ideally monodispersed. So it can be monomeric or dimeric or trimeric, but we shouldn't have mixtures of the different forms. Sample quantity. NMR is inherently a very insensitive technique, as we'll come on to. And so we need quite large amounts relative to other forms of spectroscopy. High molecular weight. As a molecule gets higher, but in molecular weight, it tumbles more slowly in solution. Any molecule, as it starts to slow down its tumbling, it has a degradation in the quality of the signal that you get back in the NMR spectrum. So there is a general rule of thumb: the higher molecular weight, the lower the quality of the data that you're going to get at the end. So we have effective molecular weight limits. We cannot look at large multi-domain proteins by NMR. They're too large for the technique. We can look at isolated individual domains. So we tend to look on the smaller side and deconstruct and then build up pictures from there. Low spin density. We need a lot of NMR active spins in a molecule in order to tell what's going on. We're saying we've got site-specific detail, and we need a lot of sites around the molecule in order to build up a picture of what the shape and properties are. Nucleic acids in particular suffer from having quite a low spin density. And speed. We've said already, the NMR data comes at a price of the amount of time that you need to invest in it to get useful information. And if you can use crystallography, you would do that in preference. So we just said that NMR sees everything that's in there. NMR does see all of the states that are present. So if you've got dynamic states in multiple conformations, illustrated in this slide, you will see effectively all of the states that are in there. And it's very difficult to tease out individual states by NMR directly. You would have to use computational methods to look at the range of motions that are occurring, and we can look at the rate at where the motions are occurring, and we can look at where the motions are occurring, but we would rely on having complementary computational techniques to work out what the individual contributing states are. So it's used in conjunction with computational methods. If we were to interpret this data in terms of a single static conformation, we would get what's called a virtual conformation, which is an erroneous, whatever, beast. And so we need to take into account that NMR sees everything that's in there, and it reports back on the ensemble of what's there. What I'm doing today is I'm introducing NMR from the first principles, and what's going to happen next in the next lecture on Tuesdays, Jane Wagstaff will talk in more detail about some of the examples that you get in real case studies in from LMB's portfolio. And so this is a slide I've nicked from Jane, and it's just illustrating the sort of approaches that we can apply to NMR experiments that will reveal detailed pictures of the dynamic processes that are going on inside the molecule. And this is the sort of motions that we're aiming to pick up in the behavior of the molecule. Jane will go into more detail about this next week, but NMR has this handle on dynamic processes of different timescales. So where does the NMR signal come from? It's useful to say that what we do is we have these powerful magnets, that you put your sample into the center of this magnetic field, this external magnetic field. As you do that, the molecule starts to separate out into different so-called spin states. Now, all of the isotopes that I've listed there have a very interesting property that they have a so-called magnetic dipole, okay, nuclear magnetic dipole, which means the nucleus of the molecule has north and south pole, formerly referred to as a spin-half nucleus. And all of these are so-called spin-half nuclei. The blue ones are virtually 100% natural abundance. The red ones there, if we want to bring these nuclei into play, we're going to have to deliberately put them into the molecule. So the naturally occurring isotopes of carbon and nitrogen are not NMR active. So if we want to bring nitrogen and carbon into play, we have to grow the sample, prepare the sample on in a growth medium that contains C13 glucose or N15 ammonium chloride as the sole carbon and nitrogen sources. Okay. The other nucleus that's up there, actually strictly speaking, is not a spin-half nucleus. It's deuterium. Is not used for bringing nuclei into play. It's actually used to substitute out hydrogen, the protium form of hydrogen, so that we silence particular areas of the molecule by putting in deuterium in place. Okay. So these are the most common nuclei that we look at. There are no NMR active isotopes that are easy for us to look at for oxygen and sulfur. There are various ions that also really are out, don't have an easy enough, certainly not a spin-half nucleus, for sodium and zinc, but we can use ionic surrogates for that. So for example, if you want to look at zinc finger structure, and immediately below zinc in the periodic table is cadmium, so we can use cadmium instead, which has a spin-half nucleus, and it gives you some information about connectivities in zinc finger proteins, for example. We can look at lithium. Fluorine is becoming more and more interesting to drug discovery chemists because you can use fluorine very productively when you introduce side chains that are fluorinated into proteins, for example. Because it has a nuclear magnetic dipole, when we put the molecule inside a magnetic field, we can get the orientation of this nuclear magnetic dipole aligned either with or against the magnetic field. So we have these two energy states. And the energy difference between the aligned and the anti-aligned state is going to be proportional to the strength of the magnetic field, directly proportional. Now, the signal strength that we get in an NMR spectrometer is determined directly by the population difference between these two energy levels, okay? This is why NMR is such a fundamentally insensitive technique, because this energy difference is so small, we're looking at a population excess of about one part in 10 to the 5 in the lower state compared to the upper state. So automatically, we're throwing away all but 1 part in 10 to the 5 of our signal. So it makes it a very insensitive technique. It's tractable for our purposes, but it does make it much less sensitive than other forms of spectroscopy. In the absence of other magnetic fields, these nuclei will then start to spin around the axis here. And they spin at a characteristic rate that is, in the absence of other magnetic fields, it is identical for every nucleus of the same type, of the same isotope. Okay? So we can put in to spectrometers of different magnetic field strength. And as we go up in magnetic field strength, we have a cost penalty. So it costs more to get the machines that are higher magnetic field. But the benefits that we get out at the end is that you gain substantially in sensitivity. And you don't just gain linearly, because it's a Boltzmann distribution between the two populations of the energy levels. We see an advantage of signal to noise going up as the power of 3/2 of the field strength. So we gain better than linearly by going to higher field strength. We get increased dispersion of signals, so we are able to pick out different signals in the molecule much better as we go to higher field strength, and we get favorable relaxation rates that we'll come on to. The disadvantage is that it costs us a lot more money. The rule of thumb is we double the price of a machine by going up in field strength by 100 megahertz. And yeah, we don't need to worry about the rest of it. What we see then is this idea of increasing dispersion. Because of the local magnetic fields that we have within the molecule, the different nuclei, so in this case, this is hydrogen nuclei, each different hydrogen nucleus produces a separate signal that is at a distinct position because it has different effects of local magnetic fields on it inside the molecule. And you can see the amount of spread of this frequency, if we plot it in absolute frequency terms, it goes up linearly with the increased field strength. So we physically resolve signals here much better at 800 megahertz than we do at 400 megahertz. So what we need to do really is, if we're comparing data from different machines, we need a different frequency scale, which is basically the frequency normalized against the strength of the spectrometer. Otherwise, everybody in the world producing NMR spectra will have a different frequency for all of their samples, and we're all reporting different things. If you normalize it against the strength of the magnet, you use this, what's referred to as the chemical shift scale. And it's a ratio of frequencies, and it's used just to normalize against the field strength, basically. What you can see is the center line of each of these signals is identical at each of these field strengths, and that's because we're plotting it on this so-called chemical shift scale for the frequency. The business end of the NMR machine is referred to as the probe, and it's a detector coil. So at thermal equilibrium, the magnetization from the samples, these nuclear magnetic dipoles, are all going to be pointing directly up the vertical axis of this NMR tube. So the NMR tube itself is a glass tube. It has 5 millimeters external diameter in this case, and we never open the NMR tube while it's in the spectrometer. So we don't get cross-contamination between components. You can take your sample back afterwards. NMR is non-destructive. As long as the sample's stable, you get the sample back at the end of the experiment. For proteins, obviously, they're not always stable, but temperature range is normal sort of biological temperatures. We can work from 0 to 60 quite happily. The concentration range that we need is somewhere in the range 20 to 500 micromolar. We would never normally recommend going above 500 micromolar for biological samples, because you get increased risk of aggregation if you put samples in at too high a concentration. So we're controlling the concentration very carefully. We have a fixed volume in this setup, and so it means that we can't just load more and more sample in to get more and more signal, because we have to consider the problems associated with concentration of components in biological systems. So the magnetization is pointing directly up the z-axis of the tube. We can give a preparation excitation pulse where we flip this magnetization from the vertical axis, we flip it into the horizontal plane, and this magnetization then starts to precess, to spin, if you like, around the central axis at a very characteristic frequency, at its resonance frequency for this molecule. Okay? And this is the basis of the signal that we record in the NMR spectrometer. So if we look at the precession of the X component, for example, what we would see is a cosine wave that is oscillating with a single constant frequency for a single spin. Okay. By running through a process that is not complicated, but it takes too long to explain it from first principles, it's really very straightforward process of Fourier transformation. We can convert this time domain signal into a frequency domain where we extract the frequency components. Now, if we had an infinitely long cosine wave like that, and we apply a Fourier transform, what we get is a delta function. It describes a single discrete frequency with infinitely narrow width on that function. But in practice, what we have is the magnetization that we've got at thermal equilibrium. We flip it into the XY plane. We record its signal in the receiver coil. And as we're recording, it's returning back to its thermal equilibrium position, its so-called relaxation process. And so it's a ringing down cosine wave. An exponentially damped cosine wave is the form of the NMR signal that we see in practice. And when we do a Fourier transform of that, we get a so-called Lorentzian line shape. So it has some uncertainty as to what the frequency of it is, but the center frequency, there we go, the center frequency of those lines is exactly the same. It's just that there is an associated line width with the Lorentzian line shape, which is describing a little bit of the uncertainty. The line width is inversely proportional to the rate at which the signal is decaying. So we can describe this exponential decay, sorry, this is not desperately easy to control that. We can describe this exponential decay by a decay constant, which is referred to in this case as T2. And the more rapidly that signal decays, the wider that NMR line width becomes. We can illustrate that here for the time domain signal for a small molecule, which is relaxing very slowly. This is a molecule that is tumbling very rapidly in solution, okay? And as it tumbles, it doesn't release its energy to the environment very easily. Molecules, as they slow down and become much more slowly tumbling, they release their energy and relax very much more quickly. So for macromolecules, proteins, and nucleic acids, they relax very much more quickly. The T2 is a lot shorter. This is the exponential time constant. It's a much smaller for a protein than it is for a small molecule. When we process that with a Fourier transform, it shows dramatic differences in line shape. Eventually, when the molecule is tumbling slowly enough, that signal becomes so broad that it becomes unusable. So this is what I'm talking about with the degradation in the quality of the signal that you get as you go up in molecular weight. So this is what is responsible for this rapid relaxation, is what's responsible for the molecular weight limits that you experience, which means we're good at looking at small domains up to a couple of hundred residues or so. But once you get above that, there is a limited amount of high resolution information that you can get from NMR. Now, I quite like this. It's a fun analogy of what's going on inside an NMR spectrometer if you're unfamiliar with NMR. So we got, this comes from Andy Darone's book from years ago, and he's got a bell that represents a simple harmonic oscillator. Now bells in practice, they have multiple frequencies and overtones and whatever. In this analogy, we're assuming the bell is just a simple harmonic oscillator producing a single resonant frequency, okay? And what we do is we ring the bell, we record the sound that comes off from that, put it through a Fourier transformation, and we extract the frequency component. If we were to put another bell in the box and shake them both up together, we would get a signal that is the summation of these two profiles. If we run that through a Fourier transform, we extract multiple frequency components in there. And we can put more boxes, or sorry, more bells in the box, more oscillators, and we get a whole series of different frequencies in this time domain signal. We put it through the Fourier transform, and we've got now a profile, a spectrum of all of the frequency components that were in the original box. This is entirely analogous to what's happening in an NMR spectrometer. So we use the radio frequency equivalent of white light, so broadband polychromatic excitation pulses across the resonant frequency range of the NMR spectrometer, and we excite all of the spins simultaneously. We record this cacophony that comes off and run it through a Fourier transform to extract all the frequency components. It's a nice analogy. So what we see, that's half of the spectrum in frequency terms, chemical shift terms, for a protein of about 10 kilodaltons. And you can see instantly the problem we're running into. There's a very limited amount of information that you're going to be able to extract because there's so much overlap in there. So what we need to do is spread these peaks out into another dimension to get better resolution. And in this particular example, it's actually exactly the same spectrum, except in this lower trace, we have got the frequency evolution of the hydrogen nucleus along the horizontal axis, and we spread them out into the vertical axis by the frequency, according to the frequency of their directly bonded nitrogen-15. So this is an amide backbone, N15 and hydrogen in a protein of about 10 kilodaltons. And what we can see is we see an individual spot in there, which turns out to be one spot for every amino acid that's in the molecule. That's an inconvenient representation of it to look at it in that particular form of plot. So universally, when we look at these, when you see them published, these data sets published, we represent exactly the same data set here, but it's represented as a contour plot where intensity of peaks is represented in contours, and we're looking directly down on top for simplicity. But you can see there is an individual spot there for each of the amino acids, and it's a correlation between the frequency of a proton and its directly bonded N15. How we make that contact between the proton and the N15, how we do the magnetization transfer, is really beyond the scope of an introductory lecture. Take it from me. If you want to know, by all means, come and see us, and we can talk through anything you want to know about it. But the spin physics is tractable. We can work it out. Okay, so what I want to look at very briefly, and we'll skip through this, niftily, is what are the sorts of things that affect the spectrum? What properties of the molecule and spins affect the molecule? This is a Hamiltonian operator. It is the angular momentum, spin angular momentum Hamiltonian for a series of spins that is in a molecule. And I don't put this equation in there to make it look complicated. I put it in for exactly the opposite reason, to make it look very simple. It's simply the sum of three individual components. Everything that we see inside an NMR spectrometer is the result of the summation of these three terms. So we'll look at each of them briefly in turn to see how it's affecting the molecule. So the first of them is referred to as shielding. And what it means is we have a cloud of electrons, not drawn to scale, and an atomic nucleus with its nuclear dipole, and a cloud of electrons circulating around the outside. That circulation of electrons around the nucleus always produces a magnetic field of its own that opposes the applied magnetic field. So we refer to this process as shielding. The more density, the higher the density of electrons that you get around the nucleus, the higher this shielding field. So this is why every spin in the molecule is going to have a frequency that is dependent upon this shielding constants that it has. Where are we? There we go. So we get a signal, and as we said before, we depict that frequency axis as the chemical shift axis. Okay, so every nucleus in the molecule will have its own different environment. This density of electrons obviously is most strongly affected by functional group chemistry. So this is why NMR is beautifully adapted to work out what the chemical composition is of molecules. So there we are. We have this landscape of local magnetic fields. So each nucleus in the molecule generates its own little profile that adds to the landscape. So everything in there has a unique chemical shift that is dependent upon the chemical composition and the three-dimensional structure of the molecule at the same time. We also get the circulation of electrons not just immediately around the nucleus, but delocalized electron clouds in aromatic systems and in pi-bonded systems, for example. And they make a big difference to the local magnetic landscape as well. It's not just the immediately local fields. The second effect is so-called J coupling, in which the information is sensed by this nucleus by the generation of local magnetic fields that are generated by electrons in covalent bonds. Okay, so the information is transmitted through covalent bonds. And so we have, for one spin, here we go, doesn't want to know. For a particular spin, it has two possible, or we've drawn it the other way around. For this spin, we have two possible orientations for the neighboring spin. This is strongly coupled between the orientation of the electrons in the bond and the orientation of the neighboring spin. So we get two different environments where the neighboring spin is aligned either with or against the applied magnetic field. Instead of seeing a single signal, we actually see two signals, depending on whether you've got the neighboring spin is aligned or anti-aligned. And this effect is transmitted through the magnetic fields of the electrons in the covalent bonds. That separation of those two is a constant. It's dependent upon the magnetic field strength of these, and it's invariant, it doesn't depend on what field strength of spectrometer you put in. Referred to as the J coupling constant. And the third influence on the spectrum is perhaps more intuitive, and it is the direct influence of nuclear dipoles. So if you've got a spin and it's close in space to a neighboring spin, you can see the effect of the neighboring spin being either in the aligned or anti-aligned orientation. And so we get these two different states. So in fact, the separation of these two spins, these two lines, is the summation of the J and the dipolar coupling. Okay, so let's look at what that means in practice. Well, for dipolar coupling, we see the distance between the amount of splitting that you get is dependent upon two terms. It's the distance between the two nuclei and the bond angle between this bond vector and the external magnetic field. So as we tumble the molecule extremely rapidly, we sample all states equally, so-called isotropic tumbling, and we don't see, all we see is an average splitting to the two. So this angular term disappears completely. If we don't have any averaging, any motional averaging, so in other words, you have a solid state sample, what you see is all possible orientations of that molecule are represented, and we lose any ability to resolve individual signals from the individual nuclei. So for example, this is the dispersion of signals that you would see normally in a high resolution solution state spectrum for a molecule. If you put the same molecule in a solid state, then we will lose all ability to discriminate between the different signals. And what is dominating the spectrum is this broad blue line that has the dipolar coupling terms in there. Okay, there are ways of eliminating that, but essentially it requires different hardware. We don't do any of it in LMB. All of what we do is solution state NMR. There is a whole field of spectrometers where people are working on removing the dipolar coupling terms by mechanical means, but it's a different technique that we don't get into here. Now, in solution, although we don't see any splitting because it's all averaged to zero, doesn't mean the dipolar coupling has disappeared, and it gives us the ability to use this so-called nuclear Overhauser effect, which occurs through dipolar coupling between spins that are in close proximity. So by close proximity, I mean roughly 3 carbon-carbon single bond lengths, okay, about 6 angstroms or there, about 3 or 4 bond lengths. So what it gives us is the ability to accurately measure what the distance is between 2 specific spins in the molecule. The other alternative that we can do is we can pick a pair of nuclei that are set at a fixed distance from each other. So, for example, just look at the NHs in the backbone of a protein. They all have the same atomic bond length. So then if we compare different dipolar couplings for different molecules, different spins along the backbone, what we will see is the angular component is reflected by the strength of the dipolar coupling term in there. So this dipolar coupling can be used either to measure the interatomic distance or the orientation with respect to the B0 field. And that's referred to as a residual dipolar coupling. And it gives us a measure of bond orientation. In the early 2000s, well, like from the 1980s, Kurt Wuttrich's group in Zurich was working on an NOE-based structural analysis of proteins. He was awarded the Nobel Prize in the early 2000s for this work. And what you take is the backbone of a single domain of a protein, and you look at the position of hydrogens in there. We have the capacity to measure the distance between individual specific pairs of spins. We build up a map of typically between 25 and 40 restraints per amino acid residue, and we feed that information into a distance geometry algorithm, and it comes up with an overall fold for the structure. It takes a long time to do it, but it's certainly something that was done for several decades. So we have, for example, if you look at a spin like that one, a peak like that one in this so-called NOE, so it's a nuclear Overhauser effect spectroscopy spectrum, what it's showing us is that peak is saying that a spin with this frequency is close in space to a spin with this frequency. And if you have the assignment of which spin is at which frequency, you can then manually get in there and determine the three-dimensional structure of the protein. In this particular case, it was about 7,000 different peaks. And the automation algorithms for deciphering this information are still not reliable enough that you could automatically assign those and publish that as a definitive structure. So we have to do that by hand, and it takes a long time to do. Realistically, the molecular weight limit for getting high resolution structures by this method is about 40 kilodaltons or thereabouts. The residual dipolar coupling, these dipolar couplings, they're kilohertz big. But if we could just reintroduce, instead of isotropic tumbling where we get no splitting term for the dipolar coupling, we introduce just a little bit of preferred orientation, then we can recover some of this kilohertz worth of dipolar couplings. Maybe we want to recover a few tens of hertz or something. So what we do is we record the spectrum in an isotropic solution where everything's rotating equally in every orientation. And then we put the sample into a different medium that is collimated with respect to the magnetic field. So you can either use acrylamide gels where you've got an asymmetric cavity that aligns with the magnetic field, or you can use a magnetically alignable medium, but it imparts a very slight preferred orientation where we got about 1 part in 10 to the 3 excess population with the protein's major axis of diffusion aligned along the magnetic field, okay? What we would gain is just a little bit of this dipolar coupling type. It's referred to as residual dipolar coupling, and it looks exceedingly underwhelming until you see it in practice. This is the residual dipolar couplings that we measure as an example on a test system here for a 3-helix bundle that we were looking at the folding of. You can see this NH pair here. The bond axis is roughly collinear with the helix axis. So each of these three helices in the three helix bundle has a different orientation, and we can pick that up very accurately for the orientation of helices in a protein. And it's a very useful restraint for structural modeling of what the protein is. We can use that in more practical terms. In this particular case, this is a homotetramer of p53. So it's 355 amino acids times 4, which is a bit big for NMR, but it has an intrinsically disordered tail of 93 residues long. And we can look at the residual dipolar coupling in the context of the full length, whilst it's attached to the full length protein here. And we can see that the experimental data in blue does not fit to a random coil for that structure. It's one of the seminal papers. It's from a little while ago now, but it's a seminal paper that illustrated if we constrain the model to have 2.5 turns of alpha helix at this point, and we have 22% of polyproline 2 helix in the C-terminal portion of this region, then we can get the experimental and the simulated data to be in very acceptable agreement. So we're using just RDCs in this case, residual dipolar couplings, to get the orientation of individual peptide planes. I'm going to skip over that one. And this one is analogous to FRET. And we can put a spin label onto a molecule. Basically, when you have a paramagnetic center, if we attach a radical, paramagnetic radical onto the protein at some point, so we can couple it, for example, to cysteine side chains, we can then get the bleaching, the enhancement of relaxation of any spin that comes within 35 angstroms of this paramagnetic center. So it can be used in terms of a spin label for working out aspects of the geometry of proteins. The key part of this experiment is that this paramagnetic radical will bleach and enhance the relaxation of NMR spins. But if we reduce it with a scorbate to a hydroxylamine, which is diamagnetic, we don't get this bleaching anymore. So you record the spectra before and after the addition of a scorbate, and you get a very good idea of what are the centers that are within in range of this paramagnetic center. So structurally, what we have is some molecular mechanics force fields. They have intrinsic terms in the molecular mechanics to constrain in a Hooke's law type way as masses on springs, where we've got idealized bond lengths, bond angles, bond torsion angles, and so on, and the electrostatic terms. And we add to that a whole selection of structural constraints. So from these NOEs, as we said, from the RDCs, we can use chemical shifts to give us an idea about constraints for modeling, and these PREs, these paramagnetic relaxation enhancement spin labels that we were saying about. And you throw all of that into your molecular mechanics algorithm. The idea is, if we want to find the structure of the protein, ideally it will be the minimum energy point along the coordinate. Now, in this case, we're describing one coordinate. In modeling a protein, we've obviously got many thousands of coordinates to simultaneously optimize. If we were just to take an energy minimization protocol, it would get us into what is likely to be a false minimum, okay? That is not the true structure. So in the process of simulated annealing, we take 100 different structures that start from different starting points along this profile, from random starting points. And if they all converge to the same point and they're consistent with the experimental data, then we can be confident that we've reached the global minimum energy structure. The point I want to make from this slide is that when you look in a database of deposited NMR structures, you don't see one single structure describing what's in solution. What you see is an ensemble from multiple simulations, and it's giving you a depiction of how well these elements converge. So this is why NMR structures contain multiple states, usually commonly anything between about 20 and 100 models depicting the consensus structure. And a fantastic advance coming from AlphaFold in the last few years, because it gives us the ability to rapidly generate, in the space of about an hour, we can generate a candidate structure that we can then use to evaluate and pick out certain NOEs without doing 7,000 NOEs for this folded domain in the middle. We can maybe do 50 NOEs that we can pick out, which is a lot quicker to do. We can do that in the space of a few days rather than having to generate the structure from de novo. This particular one, the pink one, you maybe can just see in there. The pink domain is a structure that I did by traditional NMR structural methods. The green one is the AlphaFold structure, and they almost perfectly agree. The big difference is the AlphaFold structure took about an hour, and my modeling took about 9 months to do that. And that was just a couple of years ago. So, yeah, I don't think we need to say much else about that. This is the basic idea of the typical NMR spectrum of a protein. We get the dominant feature in where signals occur is the functional group chemistry. So all of the NHs occur in a region up here. All of the methyls in side chains occur in a region at this side. If we want to get more information than that, we really need to incorporate M15 as a minimum into the backbone of the protein by growing your sample up with M15 as the sole, M15 ammonium chloride is the sole source of nitrogen. And then what we have in this typical representation, we've seen it before, we've got proton chemical shift, and the chemical shift of it's directly bonded M15 throughout the backbone of the protein. And in this particular case, 219 residues. So we get 200 and roughly 200 different spots in there that are reporting on the state of the magnetic field at each of those individual sites. So we have this site-specific ability. We can assign which of these spots corresponds to which of those amino acids, which means when we add a binding partner, if we've got any of the amino acids that are outside of the contact surface region, we see no perturbation of the HSQC spectrum as we add a binding partner, okay? But for those sites that are close to a contact surface, we get migration of the magnetic environment. And it enables us to plot the magnitude of that shift perturbation versus residue number, and we can spot where the surface is, but we can go further and we can titrate these and get a perturbation as a function of ligand concentration and use that to get accurate binding affinities for the association. It can be applied even further. So we look in this case in one of the blood clotting factors, thrombin, and it's binding to 3 different ligands at the same time. Stefan Freund did this work a few years ago. The color coding on these structures here is showing the perturbation that you get as you add in the different ligands in there. So we can get an idea of what is the structural consequence? Where structurally are we perturbing the environment as we add these different ligands? And we can do multiple ligands at the same time. We can get high resolution structures of docked bodies. So in this case, we've got a peptide binding to a folded domain of about 150 residues. What we do in this particular case, we set up a nosy spectrum with filters in there, and we reject any signal from this axis where the hydrogen is directly bonded to a C13. On this axis, we only accept signals where hydrogens are attached to a C13. It might seem confusing at first, but what it gives us in the end is all of the NOEs that you see in there are originating from an unlabeled peptide onto the surface of a protein that is C13 labeled. So all of these spots are NOEs that are coming across the binding interface. And you can then put all of those restraints, the chemical shift perturbations and the 35 NOEs in this case, with a flexible body docking procedure and get a good model of the complex. What is the molecular weight limit for NMR? Well, there's no hard and fast rule on it. It depends what information you want. Even for large complexes, we can still get information as to where we're perturbing on the surface. We wouldn't get a high resolution structure for something of this quality, but we can still extract information going up to even quite large complexes. We shouldn't get too fixated with the macromolecules. In this case, this is a DNA translocase. So we got 6 times 53 kilodaltons is the total size of this, which is way outside of NMR's capability to look at the protein. However, we can look at substrate turnover, in this case, the turnover of ATP and conversion into ADP. And we can get out just by looking at the phosphorus signal in this case and get useful kinetics information and information that what components of the complex were required in order for us to get turnover of this DNA translocase. We can do relaxation filtering. We've seen this before. A small molecule relaxes very slowly. If we were just to wait for this, so we don't start recording the signal immediately, we wait a little bit after the excitation pulse, we would have all of the large molecule signal disappear and relax away. And we've only lost about 5% of the information for small molecules. The overall profile, looking at an intact erythrocyte, live erythrocytes in this case, we got broad signals from the macromolecules, and superimposed on that is sharp signals from the small molecules. If we do the relaxation filtered experiment, we get a high resolution spectrum for the small molecules only of this, which can be very useful for metabolomic studies. There are a lot of groups working on tracing hundreds of metabolites simultaneously from the same NMR spectrum. I'll skip from there. We can estimate molecular weight and protein quality. There's a built-in quality control when we do our stuff. So for example, this is an unfolded protein. We can see in the NH region, we get very little dispersion. All of the NHs are in the same environment when this is unfolded. When you fold the protein up, they all go into a core where there's lots of different influences on the environment. So we can see at the other end of the spectrum in the methyl region, there's no dispersion for a folded protein. And yet for an unfolded protein. For a folded protein, we have good dispersion of the frequencies of signals. Instantly then, if you look at one of these plots, you can see the quality of the data that you've got in there. This one on the left is for a folded protein. This is for an unfolded protein. And when the protein aggregates, it looks like that. And NMR gives you the built-in quality control. We can label the intensity of peaks. So we know the identity of each of these peaks in the HSQC, but we can label its intensity with the dynamics of the protein backbone. And we got a whole range of different dynamic processes that can be basically coded into the intensity of the peak. We're running out of time, so we'll move on from there. Getting pKa values. Again, we can do this with site-specific detail and get individual pKa's of different side chains, specific side chains in the protein. Drug screening, we're able to identify hits for small molecules that bind, oops, that shouldn't be there. Small molecules that bind to protein surfaces and used for the early stages of drug discovery. And these are different techniques that can be used to prove whether or not different molecules in mixtures, small molecule mixtures, which of the components bind and which don't. And there are different approaches to revealing that. So that's just a summary of the whole thing. We've got protein quality control, structure and dynamics, molecular interactions, and everything is quantitative, so we can get out rate constants and dynamic processes at the end. If anybody wants to know any more details, the contact details for the NMR facility are online. Go to the NMR LMB intranet, and I'm happy to take any questions if anybody has them immediately. And if not, that's fine. Good.